All right, everyone, welcome back. So, this is where we left off last time. We introduced the major tree of life here, and we talked about the different domains of life. That was bacteria, archaea, and eukarya. Now, we're going to start today by talking about domains bacteria and archaea. So, we have a unit coming up that's all about bacteria, and we're going to dive in great detail for that unit into their anatomy and physiology. So we're not really going to talk at length about that information here. We're going to talk more briefly about bacteria, and then certainly give some attention to archaea. Now, as prokaryotes, we know that they have no nuclei or organelles. We know that their DNA is in the cytoplasm. It's not organized inside of a nucleus. No other organelle contains it. And so those processes that the cell has to do, they're not compartmentalized like they are in eukaryotes. They all happen in the same shared space inside of the cytoplasm. Okay. Now these cells, this group was first observed in the 1600s by Hook and Van Leeuwenhoek, both of them together when they were doing a lot of their first descriptions of what cells were, with looking through these early microscopes. At the time, they didn't really give them a special name. There wasn't really enough known to help distinguish these major groups at this time, and Leeuwenhoek mainly called all of these groups animalcules for quite some time. But eventually, about 200 years later, they were actually given the name bacteria. Okay, and this is a word in Greek that's derived from a word that means small staff or cane, said the same way. Now, let's look at the basic anatomy, real basic anatomy of a bacterial cell. Okay, we have a cell membrane to start with, just like everything else. All living things being made of cells has a cell membrane. Now, their cell membrane is made of a phospholipid bilayer like us. That is two layers of phospholipids that are angled tail to tail with their heads facing out into the water. Okay, into the liquid of their surrounding environment or their internal environment. And then they also have a cytoplasm. The cytoplasm contains no organelles. It contains DNA that hangs out in a central region called the nucleoid region. It's just a place where DNA is organized. It also contains ribosomes, which make proteins, enzymes that make all kinds of other things, and everything that a cell needs to function. That includes its various resources that it's gathering over the course of its life. And then on the outside of the cell, we typically see a cell wall that is a tough, rigid protective structure. This outer layer that encapsulates the cell as a whole. It's sort of like an armor, a suit of armor that it wears. It's very tough and rigid, and usually makes the cell adhere to a specific shape. And now we're going to, as we explore this unit, talk about different kinds of cell walls. And so different kinds of cell walls are made of different things. For bacteria, the main ingredient, the main thing they're made of is peptidoglycan. Okay? And in the next unit, when we talk about bacterial anatomy, we're going to dive into what that specifically is made of, what that looks like, and how it's made. How these walls are structured, things like that. But for now, that's what I would like you to know. Now, like all prokaryotes, bacteria are also exclusively unicellular. And they also reproduce asexually. Okay? That is, they don't use a partner to replicate. They copy themselves. The cell literally just divides and forms multiple new cells from that original division, from that original cell. Okay? Now, that again doesn't mean that bacteria don't work together. They can and do. A lot of them will live in like communal groups. Both with members of their own species and even with members of other species, which gets really interesting. So they're not exactly all that selfish, as you might expect. Now, bacteria can actually be quite cooperative and share resources in some cases, including their own DNA at times and specific genes. This, again, serious issue for medicine because this means that bacteria that are resistant to specific antibiotics, for example, can share that resistance with other bacteria. Or if you have bacteria that are pathogenic, they could share the disease-causing lifestyle. If it's a gene or two, they could share those genes with their neighbors and make them pathogenic, which gets very concerning. Now, these a lot of bacteria can even form biofilms, which we'll talk more about again in these future units. But like that can be really substantial too, where they can actually work together in order to survive as a group, including surviving exposure to antibiotics and sterilizing agents. Now, bacteria, like many microorganisms, are actually, we would call them ubiquitous. They occur everywhere, all around the world, in virtually every environment you can imagine. It's very rare to find a place that is actually devoid of bacterial life. And we may never even really know how many species of bacteria there actually are. We're constantly discovering new species. We're realizing forever, like continuously, that these groups are more diverse than we previously thought, which is really impressive. We're constantly discovering new species and also new strains of existing species, right? Distinct variations of a species, okay? Now, that's all we're going to do about bacteria for now, because, again, whole unit dedicated to it in the future. So we won't go too crazy on it today. We're going to talk instead more about domain Archaea, okay? On a superficial level, this group strongly resembles bacteria, okay? They are prokaryotes. So again, they are single-celled, without organelles, without a nucleus. They reproduce entirely asexually without partners. But there are some really important differences between this group and bacteria. They're just not so easily visible. It's harder to see these things, partly because those differences are in their genetics, they're in the proteins they make, and they're in the distinct physiology, the way that their cells work, essentially. So I'll give one major example right now. I mentioned previously that bacteria and also all eukaryotes have a phospholipid bilayer for a cell membrane. Okay, that looks like this. That is this phospholipid bilayer is like multiple lipids, these phospholipids that orient themselves in specific ways. So they aren't bound together like this. They're very distinct from one another, and they're held together in place by hydrophilic and hydrophobic interactions. That is the attraction to water or the avoidance of water. But Archaea have an incredibly different cell membrane. It's actually made not of a phospholipid bilayer, but of a phospholipid monolayer, where these lipids are actually bound together. They're literally bonded at this point and held together. We can see that over here in this on the right-hand side. The phospholipid bilayer very different from a phospholipid monolayer. Okay, so these differences between these groups, between Archaea and bacteria, are much more subtle and harder to make sense of just at a glance. This actually led to this group being confused for bacteria for a long time. It's relatively recent in discovery. 1977 is when this was formally recognized as its own kingdom of life, later elevated to a domain. Okay, and so the person who was mainly responsible for this is a man named Carl Woese. This is a microbiologist, New Yorker that was really influential in this field. Did a lot of research looking at these what he thought were bacteria that were methanogenic, meaning that they generated methane through their biological processes. And as he examined them, he looked at their DNA, and mainly like the RNA that they produced, and realized they were so distinct that they couldn't even be called the same kingdom of life. They were so incredibly different that they were wholly distinct and needed to be classified completely separately. In their research paper, these Woese and Fox, his collaborator, wrote, "These bacteria," quotes around bacteria, "appear to be no more related to typical bacteria than they are to eukaryotic cytoplasms," as in us. So eventually this group got its own name, Archaea, which then eventually became its own distinct domain as we learned more and more about it. And this is what that tree of life looks like, more or less. And so we are over here, eukarya right here, and then Archaea over here, bacteria over here. And you can see that Archaea is kind of put somewhere in the middle because we don't really know who is more closely related to who here. That we're not entirely sure, how this tree really is truly shaped. Like, who is descended from who? It is hard for us to assess right now. But this is the rough outline of these relationships here. Now, where do we find Archaea? Well, Archaea first got a reputation for surviving in some pretty harsh environments. We found Archaea living in a variety of habitats. One of the major places that we found them was actually in like deep hydrothermal vents in the ocean. So more than 2,000 meters down, beyond all light. So it's complete pitch dark. These photos, the only reason why they're possible is because we've sent submersibles down there with their own light source, because there is none, making it from the sun. And here in these deep reaches, we have these bubbling magma vents. These where like volcanic activity is happening below the surface, and these vents are ejecting these acidic gases at temperatures that are incredibly high, and they stand at the precipice of near freezing ocean temperatures. So 700 degrees Fahrenheit coming out of the vent right next to like 33, 34 degrees Fahrenheit. It's incredibly like extreme. Extreme is the word. And so some Archaea live here in these types of environments as extremophiles. Something that we call extremophiles that really mean lover of extremes. That they don't just survive these conditions, they don't just tolerate them, but they thrive in them. This is where they prefer to live. Some species are also very acidophilic. So they love acid. They love high acid conditions. So they're also well known for living in these caustic geysers around the world, including the ones that we have in Yellowstone. So, these environments, super acidic. They're also very oxygen poor in some cases. They have lots of hydrogen sulfide and other kinds of pretty aggressive and caustic chemicals. And the whole solution is boiling at a constant 250 degrees Fahrenheit, but these species love it there. But Archaea, it's not actually exclusively extremophiles. Some species live in soil and sediment. Other ones, they live inside of like the digestive tracts of animals. For example, we have methanogens which are these again are the organisms that break down different materials to make methane as part of their biological processes. It's the same stuff that Carl Woese was studying. They live actually inside of a variety of animals, including us, and so they're actually one of these organisms that's responsible for our farts, for example, the methane that we produce, as do, cows and pigs and other animals as well. And then we also have haloarchaea. These are salt-living archaea that are found in and mainly on our body, in places where we have a lot of salt, like on our skin. These are halophilic organisms. They're extremists that prefer high salt conditions. Ultimately, we used to think they were very extreme in like very specific ways, like extreme acid, extreme temperature, but also they live in conditions that we often think of as being a bit more tame, like the inside of our gut, for example. So in actuality, archaea are a lot more common than we once thought. Now, the next lecture that we have here is going to talk about the domain Eukarya. Now we're going to get started mainly talking about plants, protozoans, and chromists, and then we will finish up with fungi and animals.